Okay, so today we're going to talk about topoisomerase. And what are they? So topoisomerase are enzymes that release tension from the DNA. So you can see here we have a topoisomerase here releasing tension, and this is the basic DNA replication process going on with the other enzyme we're not going to get into in this video. We're just going to focus on topoisomerase. And you can see as the DNA replicates, it's kind of created a bubble, and the DNA behind it will get tighter and tighter, kind of like pulling a rope back. As you can see in this diagram, imagine this is the replication bubble, and without topoisomerases, the DNA will get tighter and tighter and coil upon itself, which is not a great thing to do because they'll obviously affect replication. So how do we, how does this enzyme work? So first of all, we have two types of supercoil. We have the negative supercoil, we have the positive supercoil, and the reason of distinction, or the way we distinct them, is by this right here. They see that the left-handed DNA strand is going over the right-handed DNA strand, so we call that a negative supercoil. And you can see here that the right hand is going over the left-handed. That's a positive supercoil. And yeah, next we have type 1 topoisomerase. This is when basically to relieve the tension, the DNA gets cut, literally gets cut by these enzymes, creates a break. By the natural tension, which everything has, the tension, it kind of loops around itself and religates. So it down one supercoil. As you can see here, that the negative supercoil is going this way from left-handed to right-handed. And when we cut it, and religate it, it's going to be right-handed, left-handed. So it's one supercoil down. Type 2 topoisomerases. These are topoisomerases that deal with two strands of DNA. They kind of like cut in one segment. You can see here it uses ATP to lock in the T segment. You have the G segment at the bottom here. And then it releases one phosphate, and the phosphate kind of breaks the upper DNA, and then it passes through the T strand through the G strand, and kind of releases that tension, or releases the two of them. This diagram here to give you a better idea. You can see here that these two DNA are kind of like linked together, kind of jungled together, and then because of the topoisomerase break, they kind of separated. So it would be a big problem if you don't have these enzymes. How do they help fight cancer? Here we have choricoma cancer. This is a type of cancer that proliferates under the skin, and it proliferates rapidly. It means it needs, and in proliferation, we obviously need DNA replication and tons of it too. This is a rapid replication. And so to bring us to the drug campotecan. It looks like this. Campotecan, which looks like this. Okay, so let's talk about campotecan. Campotecan is a drug, it's an inhibitor that mimics the DNA base, as to say, the base of the DNA. So when it's unraveled by topoisomerases, campotecan will go attach itself to the newly synthesized strand and kind of prevented it from religating. So it kind of creates a break, a double strand break. But how do we say this is a double strand break if it only break one strand? The reason for that is because we're recently synthesizing a newly strand of DNA. So that will be a double strand because it has the newly synthesized strand and the old strand, and double strand breaks are lethal to cells, would cause them to go apoptosis, which is cell death, controlled cell death. Quinoline, aka fluoroquine. This is what it looks like. It has a lot of rings, and okay, and this one works by inhibiting type 2 topoisomerases, which we learned earlier that it's causes the break in the DNA. So it goes in and kind of religates here Okay, so quinolone. It affects the ability for these enzymes to kind of religate, just like the first one with campotecan, but instead of causing a double strand break, fragments of double strand breaks, it just inhibits the ligation of the two strands, so it kind of breaks apart like that. And this is effective in gyrase, which is the topoisomerase of bacteria, which relieves the tension in bacteria. And these are my references, and thank you.